continue to do to make progress on this project. So this figure just shows the concept behind biological control of aflatoxin. So along the x-axis is the percent of an applied atoxigenic strain, in this case it's AF36, and then along the y-axis you have the amount of aflatoxin. And this was one of the first demonstrations that as you have more displacement with the atoxigenic strain, you get less aflatoxin in the crop. And this has since then been developed, and several biocontrol strains have been put to use in corn. But there are a variety of different strains, genotypes of Aspergillus flavus out in the environment. And they're extremely genetically and phenotypically diverse, and this just illustrates that there are many different genotypes. The top figure shows the frequency of different genotypes within a single field. And then the bottom figure just indicates there's a lot of variation in the amount of aflatoxin produced by Aspergillus flavus, ranging from no toxin production in atoxigenic strains to highly toxigenic. And the way that in the past we have really characterized the genetic variability in Aspergillus flavus populations has been through vegetative compatibility analysis, which is a culture-based technique. And the details are not extremely important, but this just shows the process. It's a long process, and it takes up to 3 weeks to get results in terms of what genotype you have. And so what we do is we pair isolates. One is a known genotype, and the other is an unknown genotype or vegetative compatibility group. And they're non-nitrate ulysin mutants. And when they grow in this media and they come together, if they're in the same vegetative compatibility group, you get a line which indicates wild-type growth when they complement each other. So this has been important for assessing the diversity of Aspergillus flavus in conjunction with evaluating aflatoxin production and identifying atoxigenic strains. And using these techniques, it's possible to identify atoxigenics for potential use in biocontrol throughout the world. And this just indicates some of the places where biocontrol has been developed through isolation and characterization of atoxigenics. Many of these are areas where the CODI lab has collaborations, but there's always a need to find more and better atoxigenic strains for biocontrol. And that's really been the focus of a lot of the work in Dr. Cotty's lab. But my particular focus as part of this project funded by AMCOE was to look at biocontrol strains for Texas. So what we did is we evaluated 16 different atoxigenic strains that were isolated from corn throughout Texas from all the different corn growing regions. And we conducted field trials in commercial fields to look at how good each of these atoxigenics were at displacing toxin producers and reducing aflatoxin contamination. And we looked at both movement up to the crop and in the soil. And a lot of what I've been doing over the past year is looking at ways to track the isolates using DNA-based molecular assays as opposed to the culture-based techniques, which are very time and labor intensive, and also looking at different laboratory assays that can assess the compatibility of these different atoxigenics and be used as an initial screen to determine which atoxigenics may have the best potential as biocontrol strains in the field. And our ultimate goal is to develop mixtures of atoxigenics that can be made into a new biopesticide product for use by corn growers in Texas. And then, of course, a longer-term goal is to really do the same thing for all the different areas where we want aflatoxin biocontrol. Our reason for wanting to do it in a region-specific manner is both so that we're not introducing exotic strains to an area, and also we assume that isolates or genotypes that are native to an area will be better adapted and hopefully more competitive in the environment. So we started this project in 2011, and we put out 16 Atoxigenics. What we used is we had sorghum grain, which was colonized with each of the 16 isolates, and we put out equal amounts of each of those atoxigenic isolates. And we had relatively small plots in cornfields, both north of Dallas and south of Dallas in 4 different locations. And this was to do an initial assessment of how effective each of these 16 atoxigenics were in moving up to the crop and displacing toxin producers. These are the results of this field trial, and on the left it shows the control and the distribution of the S strain, which are the highly toxigenic Aspergillus flavus strains. And then the purple is the rest of the Aspergillus flavus community, which includes both atoxigenic and toxigenic groups. And finally, the blue is the frequency of the applied atoxigenics. So that's the total of the 16 applied atoxigenics that were identified from corn samples taken from those fields at harvest. And in the control, we had quite a bit of the highly toxigenic S strain. It was around 20% of the total Aspergillus flavus on the crop. And we also had high toxin. But in the treated samples, we had a lot of displacement by the applied atoxigenics. Close to 75% of the total Aspergillus flavus on the crop in those plots was comprised of those 16 atoxigenics. And among those 16, this shows the frequency of each of them in both the controls and the treated plots. And you can see there's a lot of variation in how successful they were at moving up onto the crop. And we wanted to only select those that were very good at moving onto the crop and displacing toxin producers. So from this group of 16, we narrowed it down to 8, which are indicated with stars. And our goal is to have the ones that were the best at moving on the crop and being competitive as compared to the toxigenic Aspergillus flavus. We also wanted genetic diversity. Our goal is to come up with biocontrol mixtures that are genetically diverse, so they're more likely to be adapted to variable environmental conditions and agroecosystems. And we also, let me go to the next slide. We also wanted them from a variety of regions. So the second column shows the origin of the isolates. And here I just indicate whether they were from North Texas or the Rio Grande Valley. And so this again will ensure that they're adapted to a wider range of conditions. And they had different mechanisms of atoxigenicity. Many of them had a full deletion of the aflatoxin cluster. Others had partial deletions or just the SNP, which is what causes the biocontrol strain A36 to be atoxigenic. So we have a lot of variation here, yet these are all very competitive in corn. So by early 2013, we had these 16 atoxigenic isolates characterized, and we selected the 8 that we wanted to study further. And in 2013, we put out a field trial with much larger plots, so we could really look at, on a larger scale, how these different strains are performing. And also, we had greater separation between the controls and the treated plots so that we wouldn't get so much movement of the treatments into the controls. So this was a much better evaluation of these strains compared to our previous trial. And so we put out the two mixtures. Each mixture had 4 different atoxigenic isolates, and those are indicated by the blue and the pink squares on the map, which are the different plots. And then we collected the corn grain at harvest, and we looked at the displacement of other Aspergillus flavus by these applied strains. And these are the results. This was based on the culturing, the vegetative compatibility analysis. And in the controls, we had a lot of S strain, which again is the highly toxigenic Aspergillus flavus, and quite a bit of other Aspergillus flavus, but we didn't see any of the applied strains in the controls. On the bottom left are the results for mixture 1, and in these samples we had no aflatoxin, and we also had a lot of displacement of the other Aspergillus flavus population by the applied strains, as you can see by the proportion of that chart that's blue. However, with mixture 2, we still had quite a bit of toxin in the crop, and we had only about 25% displacement by the applied atoxigenic strains. So this mixture did not perform well at all. And we're not sure exactly why, but I'll show some other data that suggests some reasons why it may not have been as effective as the other mixture. For mixture 1, this is the distribution of the different applied strains in each of the replicates in the trial. And there was quite a bit of variation, but overall, each of the 4 isolates was made up about the same amount of the total Aspergillus flavus population. They're all close to 25%. However, in some replicates, we had really dominance of some isolates and didn't see much of the other ones. And so this may suggest that there were different conditions throughout the field that favored some isolates over others. But overall, we had, from the mixture as a whole, very good displacement of toxin producers. So this may suggest one benefit of mixtures is that even if one particular isolate in the mixture doesn't do well, you can have others that are going to be effective in competitive displacement and aflatoxin biocontrol. So I just want to reiterate that many of these analyses were done with vegetative compatibility analysis, which is very time and labor intensive. Dr. Putty has a very large lab with lots of undergrads working on this, and it still takes them many months to complete these analyses. So one thing I wanted to do as part of this project was to develop a faster alternative using a DNA-based assay. So instead of looking at going through the whole process of doing isolation and subculturing and these vegetative compatibility analyses, what I've done is actually isolate DNA from the spores or from the corn kernels to look at the fungal DNA. So this diagram represents the DNA. And then it's amplified by PCR, but we still have, as indicated by the red and blue, we can look at the proportions of the 2 different genotypes in the mixture. And then what I've done is measure the frequencies of the different genotypes or VCGs using detection of isolate-specific single nucleotide polymorphisms, or SNPs, using pyrosequencing, which I'll explain further. So pyrosequencing allows us to look at polymorphisms within a strand of DNA and measure the proportions of that polymorphism, as shown here. And that polymorphism, if we select the proper position, indicates which genotype you have. So for example, it shows VCG1 has an A for the nucleotide at the first position, and VCG2 has a T. So when you measure the proportions of these two nucleotides, you then know how much you have of each of those genotypes. And so this basically gives us the same results as the vegetative compatibility analysis, but it's much more rapid and precise. And so this is just to explain further how the pyrosequencing works. As the strand is sequenced and nucleotides are incorporated into the DNA strand, a reaction occurs that releases a light signal, which is detected by the pyrosequencer. And these peaks represent the intensity of the light, and they are proportional to the amount of that base in that position. So it's the relative peak heights that we're measuring at the SNP, and that tells us how much we have of each fungal genotype. And this is just a comparison of the two methods. And I just want to make the point that we can isolate DNA, do the PCR and pyrosequencing within a day or two, whereas to get results for the vegetative compatibility analysis, it takes a minimum of 3 weeks. So the challenge with doing the pyrosequencing is we need to have VCG or isolate-specific SNPs, which is relatively easy to do if you have a relatively simple mixture of isolates. And here it just shows 4 different isolates, and the colored bases indicate where there's a nucleotide that's unique to a specific isolate. So you can see there's several throughout the sequence. But when you look at a more complex mixture, which is more representative of what we have in the environment, it's much more difficult to find unique SNPs. However, we have a lot of isolates that have been isolated from corn and other crops in Texas and Arizona over many years. So we have a good idea of what genotypes are out there in the environment. And we were able to sequence the most common toxigenic and atoxigenic VCGs from Texas, along with our the atoxigenic VCGs that we're currently evaluating, so that we could look for SNPs to design these pyrosequencing assays for quantification and tracking of different isolates. However, a lot of these isolates are closely related, and it's really difficult to find unique SNPs that can be used to track and quantify these isolates. And so, in conjunction with another project, one of the postdocs in Dr. Cuddy's lab did whole genome sequencing of all of our target atoxigenic isolates, along with those other common VCGs I showed in the previous slide. And from this, we got over 500,000 SNPs, and approximately 95% of them were not unique, so they were shared by 2 or more of the isolates. However, 5% were unique, and that still leaves us with 25,000 potential SNPs that can be used to design these DNA-based assays to track the isolates. And we just finally got all of this genome sequence data assembled and analyzed, so I'm still in the process of validating these pyrosequencing assays, but we have the SNPs, so we can do it. And the next step, and what I'll do in the next few months, is I will take DNA from those same corn samples that we did the vegetative compatibility analysis on. I'll isolate the DNA and measure the frequencies of the different atoxigenic VCGs using these pyrosequencing assays that will allow us to compare those results to what we got from the culture-based results. And just for comparison, and then our plan is to, in future studies, use these DNA-based methods to more rapidly track isolates and measure their frequencies in field trials. Another portion of this project was to develop and evaluate the utility of laboratory-based assays to screen atoxigenics for effectiveness in biocontrol. And in this slide, I just want to explain our methods. So I inoculated corn kernels with mixtures of isolates and then isolated DNA from both the spores from the surface and then also from the kernels, which that's the DNA inside the kernels is what the fungus is actually infecting and producing toxin within the corn. And this just shows from previous studies we know that how well it reproduces and the extent to which an isolate infects the kernels is not necessarily the same, and so this is our justification for analyzing those two components separately. So this work was done mostly by a technician, Pummy Singh, who I supervised. And what we did is we co-inoculated corn kernels with each of these 16 atoxigenics, and then along with a common toxigenic isolate, which is CG136, so this is the most common toxigenic VCG in Texas. And we looked at how well each of the atoxigenics displaced the toxin producer. And all of them did fairly well. There was some variation, but not as much as we've seen among some other isolates in the past. And the first four on this chart show the atoxigenics that were in mixture 1, so the mixture that did best in our 2013 trials. And the next four are those that were in mixture 2, and then the last four, I guess there's only 12 in this figure. The last four were the worst performing VCGs in the 2011 trials. So these ones that performed poorly in the field did not do as poorly in this assay as we might expect, but it is interesting to note that in mixture 2, one of the isolates was not very good at displacing the toxin producer. And this is also reflected in how well it performed in the field. This mixture did not perform well. So this just shows that we can't necessarily select the best isolates using this assay, but we might be able to screen out those that are not very likely to perform well in the field and that have the least potential in biocontrol. One of the other assays was looking at the effect of temperature on competition among mixtures of isolates. And I'll just show a few examples of the experiments we did. So we co-inoculated corn and incubated either 20 degrees Celsius or 30 degrees, and then measured the frequencies of the different isolates that were inoculated. And then we looked at both competition during seed infection and sporulation on the corn kernels. And these are the results for a mixture including the two best performing atoxigenic VCGs from the trial in 2011 and the two worst. And there is some effect of temperature on how well these isolates perform. For example, for sporulation, the first isolate indicated did a lot better at 20 degrees than it did at 30 degrees. And then the last one for kernel infection, it did better at 30 degrees compared to 20 degrees. So what we're trying to get at is see how under different environmental conditions that might be present in the field, how these different isolates will perform, and select either individual atoxigenics or mixtures of atoxigenics that will perform under the largest range of environmental conditions, so that we're sure to get that effectiveness of the biocontrol, no matter what environmental conditions are present in a particular season. And these are just some examples of some other mixtures. The top shows mixture 1, some of the isolates represent mixture 1, and the bottom is mixture 2. And for mixture 1, all of the isolates performed pretty much equally well at each of the temperatures, and among isolates they were very similar. So this is a good mixture in the sense that no one atoxigenic isolate has a particular advantage over the other, so they're all going to perform well in the field. Whereas for mixture 2, we had dominance of some isolates depending on different temperatures. So this might be a problem in the field because we don't really want the applied biocontrol strains to be competing too much with each other, and this might reduce their efficacy at competing with the target toxigenics in the field. So mixture 1 had really good performance in 2013. We're going to move forward with this mixture and develop it as a biocontrol product for corn growers in Texas. And a lot of this is going to be done in the CODI lab, and I'm going to continue to collaborate with them in tracking these isolates in field trials. So with the genome sequencing, we identified useful SNPs for designing these DNA-based pyrosequencing assays. And as I just said, we'll use this in the future to detect frequencies of the atoxigenics as we evaluate them for efficacy. And the laboratory assays did have some utility in screening out which isolates and mixtures of isolates may not be the best for the field, but it's obvious that we need to continue to do field evaluations, though this may offer us a way to rapidly screen out atoxigenics that really aren't worth looking at in field trials, and this will reduce the time and effort required to evaluate and identify the best atoxigenic strains. So with that, if there's time, we can take a few questions. Yes?
Usually you have 25,000 SNPs. What's your plan for how to use those 25,000 SNPs for the bioassay?
So, in addition to having SNPs, we need, to design a good assay, we need a region of sequence where there's a conserved region so we can design primers that are going to amplify everything.
So what's the plan? You still use the PCR or you use the SNP array? Is that SNP array for the genotype?
Oh, so the pyrosequencing assays that I described, they utilize PCR to target the region where the SNP is, and then the actual pyrosequencing reaction quantifies the proportion of that SNP. So that is, does that answer your question?
I'm just interested, you have 25,000 SNPs. How to use what kind of SNP array, for other crops. You have something array for genome. You basically, even with also genome, those different methods.
Yeah, so for the purpose of this, we're not going to utilize all those SNPs. Even though in the future we may use it for genotyping in other systems. But for what I'm doing, just to track the frequencies of specific atoxigenics, I'm just taking out the most useful SNPs that we can design a good assay for to actually do this quantitative pyrosequencing. Yes?
I didn't hear what the rate of application was for those 4 strain mixtures. In your big plot test, what was the rate of application for those?
Yes, it was 10 pounds per acre, I believe. It was that may not be right, I think it is though. We did the same rate that the AF36 is applied commercially.
Did you check any of those for cyclophysic acid production, those states?
Yes, and most of them did not produce it, and I believe 2 of them did. And that really, we have been targeting, trying to find isolates that do not produce CPA.
Last question, your frequency of isolation, was that from kernels?
Yes.
And so that totaled 100% among the 4 that you applied, right? But I guess the question is, what was the colonization by how many kernels were not infected by those 4? What was the, you see what I'm asking?
Yes. So we did not evaluate that, or if we did, I haven't seen that data. Yes.
One question. How would you characterize in your lab assays? Okay, because I can sense it's mixed, the results are somewhat mixed. And I'm a little concerned about taking some lab assays that are mixed and then using that to assess performance out in the field. You know what I mean? To me, you still got a lot of work to do in that regard. I'm not sure I would hang my hat on some very mixed results there.
Right, and I agree. And we have a few other assays we're still working on the results for. But basically what we concluded is that field evaluation is still very important. And we had some promising results initially with laboratory assays, really confirming some field-based data that we had. But I think overall you're right, it has mixed results. So I think we may, to an extent, move away from that, and especially once we have the rapid DNA assays to track isolates in the field, that's probably the direction that we'll focus on in the future. Yes?
I have a question. So you mentioned that at this stage you're developing maybe a Texas-focused set of isolates, and then apply a similar sort of methodology in other areas. What's your vision for the scale? So are you thinking each state, or do you have regions in mind, like a Texas, Arkansas, Louisiana, or where do you think?
So we're hoping to do region-wide, so in a particular area, but our approach is to go into other states and see if we have these same atoxigenics out in the environment. And hopefully we can do that relatively rapidly with these assays and some other confirmation. So if we find particular genotypes that are widespread in the region, then we can make just a single biocontrol mixture product for that region. So that's really our goal. Yes?
I have 2 questions. The first one relates to how stable the A proposed are in terms of producing the toxin. And the second one also, second question relates to certification what the rules are. There's a lot of volatility in terms of performance of the food So how do you anticipate sort of trying to start some of those observations, trying to perhaps using a standard approach, try to look at commonalities of what financial factors COVID can be? Because I mean, they are highly variable, the performance, okay. So how do you go about, do you think you're going to go about trying to sort of set by example those things we are ready to see about it?
Right, so to answer your second question, we want to find mixtures and develop mixtures where we won't necessarily have all of the VCGs perform really well in all areas, but as a mixture as a whole, you can apply the same mixture of isolates, and you'll at least have one or more of those isolates being highly effective in that area. So that's why we're really looking for genetically diverse, phenotypically diverse atoxigenics to put in those mixtures so that they will perform over time and space, that as a whole they will perform well and be effective. And your first question was, how stable is the atoxigenicity? So for the ones that have the full deletion, we expect that to be very stable. They're missing the aflatoxin cluster, so we expect that to be very stable. For the SNP, we know from work with AF36 that SNP has been stable. We had samples that included isolations over a period of 20 years of AF36 from the field, and that SNP has been very stable. So we expect that to be stable as well. And we do look at those things when we're assessing these atoxigenics. Yes, sir.
Looking back on this past year, looking forward to the next year, what did you learn from this past year that you think will refine your purpose and your objective, in achieving your objective this next year with this result?
So, well, I think that we learned that, one, field assessments continue to be very important, but the main thing we need, the most important thing, is having a faster way to analyze these samples. So we're hoping that we can increase the extent to which we do field trials and field sampling using these rapid DNA-based methods in the future, and really have that be the focus of future evaluations for developing these biocontrol mixtures for Texas and other regions.